Hi, everyone. Welcome to session 1 of this meeting on the role of the microbiome in HIV transmission. We have 3 exciting invited speakers for you today, and they're going to cover topics ranging from the role of the vaginal microbiome in transmission, the rectal microbiome in transmission, as well as using an SIV infected macaque model, again, to look at transmission. We're going to have 3 talks back-to-back, 20 minutes each, followed by a discussion session where we hope we'll get questions from you guys for us to pose to the speakers. So all 3 talks will have questions addressed in that discussion session at the end. So without further ado, I'd like to introduce our first speaker, who is Adam Burgener, who is a professor at the Center for Global Health and Diseases at Case Western Reserve University. The central theme of Dr. Burgener's research program is to understand host and microbial mechanisms that are important in mucosal immunity and susceptibility to disease. And he's going to talk to us today about the impact of hormonal contraception on the vaginal microbiome and HIV acquisition.
Thank you to the organizers for inviting me to speak today. Today I'll be discussing the vaginal microbiome and HIV acquisition and its interactions with hormonal contraceptives. I have no disclosures. So it's always nice to start a talk with a nice picture. What we're looking at is a picture of the gut of a mouse and the microbiota sitting on top in purple and red, sitting on top of a thick mucosal layer in green, atop of epithelial cells in blue. And when you look at this picture, you can appreciate a couple of things. One, that we're vastly outnumbered and that bacteria are living in all surfaces of our body. And also that the microbiome is in very close contact with mucosal surfaces, and these interactions are very important. The microbiome is very important for our health, as we all know. And one concept that has been linked to many chronic diseases is the concept of microbial dysbiosis. And microbial dysbiosis can be characterized in many different ways, including changes in metabolic functions, the distribution of its members, or its diversity. And microbial dysbiosis has been linked to increased risk of chronic diseases such as cancer, diabetes, obesity, as well as cervical cancer, preterm birth, and increased risk of sexually transmitted infections. And microbial dysbiosis in both of these compartments, the gut as well as the vagina, have been linked to increased HIV pathogenesis and transmission risk. Today, we'll be talking about HIV transmission, and a very important microbiome for HIV acquisition in women is that of the vaginal microbiome. And the vaginal microbiome can be divided into 2 very broad groups. Those that are dominated by Lactobacillus species, including Lactobacillus crispatus, Lactobacillus inners, Jensenii, and others, which can vary considerably in their inflammatory properties as well as their ability to produce lactic acid. And then women that do not have lactobacillus instead have anaerobic bacteria, including Gardnerella vaginalis, Atopobium, Prevotella, Mobiluncus, and other facultative species. And this is commonly associated, this is what's known as microbial dysbiosis or bacterial vaginosis. Now, in order for HIV infection to occur, it must traverse the many physiological and biological barriers of the female genital tract, including a squamous epithelial barrier, a layer of protective mucus, as well as resident immune cells. However, a major risk factor for increased HIV acquisition in women is genital inflammation. And this can be broadly characterized as increased pro-inflammatory cytokines, increased number of activated T cells, or the number of T cells, which are targets for HIV, and epithelial barrier disruption. Now, the vaginal microbiome gives many functions that are important for mucosal homeostasis. Lactobacillus species can produce lactic acid, which help to keep the vaginal pH quite low and acidic. It also provides some other antimicrobial functions, and lactic acid itself can be anti-inflammatory. Whereas in the absence of lactobacillus and the presence of bacterial vaginosis, this is associated with many changes in the vaginal microenvironment, including increased vaginal pH, pro-inflammatory cytokines, epithelial disruption, short-chain fatty acid production, as well as the production of enzymes by the bacteria, which can degrade protective features, including mucins and mucosal IgA. So it's not surprising to know that bacterial vaginosis has been associated with increased HIV acquisition risk in women for a long time. With the advent of better molecular tools, we've gained a better appreciation of the relationships between the vaginal microbiome and HIV acquisition. And this is a study showing data from the FRESH cohort in South Africa, looking at the microbial diversity in young women. On the top right, we can see the plots. Some women had Lactobacillus crispatus in yellow. Many had Lactobacillus inners in green. Some women had Lactobacillus had Gardnerella vaginalis in blue, and those in red were highly polymicrobial with no predominant taxa. An important observation here was that women with polymicrobial microbiomes were at fourfold increased risk of acquiring HIV than women with L. crispatus. And this was associated with increased levels of pro-inflammatory cytokines, as well as increased activated T cells in the vagina, and this is shown on the bottom right. Follow-up studies have also shown a dose-dependent relationship between anaerobic bacteria and increased HIV acquisition, including Gamella, Dialister, Parvomonas, and others, which independently associated with the 2 to 3-fold increased acquisition risk, whereas Lactobacillus crispatus and to a lesser extent Lactobacillus inners is associated with relative protection from HIV. Our group, in collaboration with CAPRISA and University of Washington, performed a sub-study of women that were enrolled in the CAPRISA 004 trial, which tested a vaginal microbicide for HIV protection. And the major finding was that women with vaginal lactobacillus had the drug work 3 times better than women that did not have vaginal lactobacillus. And this was associated with drug metabolism by anaerobic bacteria, as well as increased genital inflammation. However, follow-up studies showed that from the Partners trial from Renee Heffron and Jared Baton showed that there was no relationship between bacterial vaginosis and the efficacy of oral PrEP, suggesting that the modality and delivery mode of pre-exposure prophylaxis is important for whether these interactions may occur. There are over 800,000 new HIV infections in women around the world, and many of these are in sub-Saharan Africa. And the prevalence of bacterial vaginosis in many countries in Africa is quite high, and this is shown on the pie charts on the right. In purple shows women that have bacterial vaginosis, and in orange and yellow are women that have vaginal lactobacillus, showing that BV is quite high. Therefore, the promotion of lactobacillus could have a significant impact on HIV acquisition rates for women, as well as other reproductive health outcomes. Other factors also affect HIV acquisition risk, and this is a meta-analysis that shows a relationship between injectable hormonal contraceptives such as depot-hydroxyprogesterone acetate and relative risk of HIV acquisition compared to other contraceptives. In these various meta-analysis, an observation was that DMPA is associated with 1.3 to 1.5-fold increased risk of acquiring HIV compared to oral contraceptives or no contraceptives. However, these studies were post-hoc analysis of from various cohorts, which were confounded by other variables which were not controlled for in the original study design, as they were not designed to study this specific question, which led to the ECHO trial, which randomized women to 3 different contraceptives, including DMPA, copper IUD, and an LNG implant, which found that there was no difference in HIV risk of the different contraceptive methods between the different contraceptive method groups. However, it should be noted that this study trial design was designed to detect a 50% or greater increase in relative risk of acquiring HIV. So with these observations, there has been an abundance of papers that have looked at the effects of DMPA on the vaginal mucosal microenvironment. And this is a review that has been submitted by my student, Hosanna Ayeli. And she went over papers over the last 20 years looking at the relationship between DMPA and the vaginal microenvironment. And one of the questions that came up is, how does DMPA associate with changes in the vaginal microbiome? And while some studies showed a decrease in the relative levels of G. vaginalis or Lactobacillus inners or some of their functions, other studies saw an increase in G. vaginalis or Lactobacillus inners, whereas a majority did not find any significant changes in the microbiome, suggesting that these relationships are complex and there are no clear associations. However, studies have found that, many studies have found that DMPA associates with increased cervical vaginal mucosal immune cells, including CD4 positive T cells that are activated, as denoted by these various publications. Also, DMPA has been associated with changes to molecular pathways in the vaginal epithelium. And many papers have shown that this associates with changes in pathways related to epithelial differentiation and tight junction formation. And a picture is worth 1,000 words, and this is beautiful work done by Dr. Ann Carias at Northwestern University, where she studied the vaginal epithelium in pigtail macaques over the menstrual cycle, as well as when the pigtail macaques were on DMPA. And as you can see in this image, with DMPA, there is a loss of the thickness of the vaginal epithelium, loss of the stratum corneum, which is more pronounced than that of the luteal phase when progesterone levels are high. And this is associated with increased levels of HIV target cells within the tissues of the vagina. So with all of these observations, we thought that the interactions between the vaginal microbiome and hormonal contraceptives may be important for HIV acquisition risk. And a recent study by Haddad et al. Indeed found an association between BV and HIV acquisition, in which women with BV who were on DMPA were at increased risk of acquiring HIV compared to women with BV and were not on DMPA. So we have been partnered with HIV prevention trials and leadership in the HIV prevention field to study vaginal mucosal biology and how this relates to HIV transmission. And this includes Connie Seelam, Jared Baton, Nellie Mugo, and Jay Lingappa from the Partners Prep trial, Sharon Hillier, Jeannie Morazzo, and Mike Cherenje from the Voice trial, and Salim Karim and Koresha Karim and Lyle McKinnon from CAPRISA. And with their generous support, we have been generating data to understand these relationships. So there's different ways that we can measure the microbiome, including looking at using target analysis to look at genes, which can give us compositional information of the microbiome. We can also do shotgun sequencing to look at the metagenome, which gives us functional potential of the microbiome. By looking at transcripts, messenger RNA, by metatranscriptomics, we can understand the functional potential. And by metaproteomics and metabolomics, we can look at translated protein products as well as the secreted metabolites, which gives us better information on the functional potential of the microbiome as these later components can interact with the host. And our laboratory uses these later techniques to look at the vaginal microbiome. So we have profiled over 1,600 women by metaproteomics techniques, and this is a diversity plot, and each dot represents a specific woman, and you can see that in the distance between the dot is how different compositionally they are from one another, and including those who had L. crispatus, Jensenii, or others in the top there. On the bottom right in brown are women who have Lactobacillus inners, and the bottom left are women who have G. vaginalis, and in those in red are women who have polymicrobial microbiomes with no dominant taxa. So we did a case control analysis within the CAPRISA 004 trial, as this microbiome data had been published previously. On the right, women who are using DMPA compared to women who are using NET-N or oral contraceptives, there was no difference in the relative risk of acquiring HIV. When we stratified by microbiome type, in non-lactobacillus dominant women, we did not find any difference between HIV acquisition rates and contraceptive usage. However, in lactobacillus dominant women on the left, we saw a threefold increased risk of acquiring HIV in women on DMPA compared to NET-N or combined oral contraceptives. Using baseline rather than visit data did not change these observations. And it should be noted that within the CAPRISA 004 trial, the distribution of contraceptive use is shown on the left there, where the majority were on DMPA, while others were on net N or oral contraceptives, while a small minority were on non-hormonal contraceptives. And this is the plot of the vaginal microbiota of each participant in CAPRISA 004 on the top, including those women who have L. crispatus on the left, Lactobacillus inners made a large proportion, those with Gardnerella vaginalis, or those who were polymicrobial with no dominant taxa. And there was no association between contraceptive usage and the proportion of Lactobacillus dominance or non-dominance. In the PCA plot in B, we saw a complete overlap between different contraceptive types and microbial composition between DMPA, net N, and oral contraceptives. We did, however, observe a slight decrease in the diversity index of women using DMPA compared to oral contraceptives, and to a lesser extent net N, as well as a slight reduction in the amount of Gardnerella vaginalis or megasphaera in women using DMPA. However, adjustments for these did not change the primary observation with HIV infection. We hypothesize that there may be changes in the vaginal microbial functional capacity of Lactobacillus species in Lactobacillus dominant women that may contribute to this observation. So by profiling the functional pathways at the proteome level, we saw no changes in the distribution of functional pathways involved in metabolism or other functions in women using DMPA, net N, oral contraceptives or non-hormonal contraceptives. So we then measured MPA levels in serum by targeted mass spectrometry, and this work was carried out by Rafilwe Molothagi from CAPRISA, as well as Lyle McKinnon and Al Leslie at the African Health Research Institute. And by comparing MPA levels in Lactobacillus dominant women, we saw a relationship between MPA levels and mucosal proteome changes, as shown on the left volcano plot. Whereas we did not see changes in the mucosal proteome when stratified by high MPA versus low MPA on the right. These mucosal proteome changes were associated with increased pro-inflammatory pathways, as shown in B in Lactobacillus dominant women, including ERK5 signaling, chemotaxis, NRF2 oxidative pathways, as well as glycolysis and gluconeogenesis, suggesting that inflammation was different between Lactobacillus dominant women by MPA levels. This plot shows a clear relationship between MPA levels and separating Lactobacillus dominant women by MPA levels. So in brown, in the brown plot in B, you can see, and in yellow are Lactobacillus dominant women, and they were separated by MPA levels. Whereas in the purple plots, there is no separation in non-Lactobacillus dominant women. Now, the protein biomarkers in C are the proteins which comprise the majority of the variance between the proteome of high MPA versus low MPA. And these biomarkers are involved in metabolism and inflammation pathways. And right beside it, many of these biomarkers are independently associated with increased HIV acquisition risk, as shown in red, including PYGL, PGLS, and ALDOC. In D, you can see a dose-dependent relationship between these biomarkers and MPA levels in serum, which increase, whereas in purple, these biomarkers were already at high levels in non-Lactobacillus dominant women and were not modified by DMPA or associated with MPA levels. We then compared inflammatory pathways in the mucosal proteome as stratified by different microbiome groups, as shown by the activation scores. And each one of these dots represents different non-Lactobacillus dominant microbiome types, including polymicrobial microbiota in red. Purple are those dominated by G. vaginalis, and those in brown are those with Lactobacillus inners in comparison to those dominated by Lactobacillus crispatus. And you can see, as denoted by the squares, many of these pathways overlapped with MPA levels, including LXR, RXR activation, acute phase response signaling, and NRF2 oxidative stress response, suggesting that inflammation pathways may be in common between those women that have microbial dysbiosis, or they may converge at some point. And this is work recently presented by Renee Heffron and Heather Jaspin, who are leading a mucosal substudy of women in the ECHO trial, looking at what happens at in mucosal inflammation profiles of women 1 month post-initiation of different contraceptives. And this was a specific observation that was presented last year at IAS, where women using DMPA had higher levels of Th17-like cells in the vaginal mucosa compared to women using LNG implant or copper IUD. It is unknown whether these inflammation profiles are linked with increased acquisition risk or are related to the microbiome, and these studies are ongoing. So in summary, vaginal microbiome alterations underlie increased risk for HIV acquisition in women, and both the vaginal microbiome and DMPA associate with mucosal alterations related to inflammation, epithelial barrier disruption, and recruitment of HIV target cells. So we believe the vaginal microbiome context may be important for DMPA-associated DMPA genital inflammation and the risk of HIV acquisition. However, it must be noted that this study, CAPRISA 004, has many limitations, including that this is a post-hoc analysis. The study groups were not balanced. It was not randomized for different contraceptives, and there are differences in risk behaviors that may not have been captured in the primary study. So nevertheless, we believe that further study to understand these microbiome host interactions with hormonal contraceptives are important for women's health. With that, I would like to thank my lab, especially Laura Romas and Michelle Perner, for performing much of this analysis and this papers presented today, as well as Hosanna Ayeli, as well as our important collaborators, Salim Karim, Karisha Karim, and Lyle McKinnon and Rafilway, as well as Al Leslie, and our other collaborators which contributed to the study, as well as the participants in the CAPRISA 004 trial and our other study cohorts. Thank you very much.
Hello, good evening from Madrid. It is my pleasure to introduce Dr. Colleen Kelley. Dr. Kelley is professor at Emory University School of Medicine, the Division of Infectious Diseases, with previous clinical experience in HIV and sexually transmitted infections. She leads a translational immunology lab focused on human mucosal susceptibility to HIV infection, with specific expertise in flow cytometry and explant HIV challenge experiments with mucosal tissues and the microbiome. She will present her fascinating work focused on the influence of the rectal mucosal immune environment on HIV acquisition risk.
All right, well, thank you so much for inviting me to present our data. These are my financial disclosures, and I will go ahead and get started on telling you a little bit about the rectal mucosa and why it's important in understanding HIV transmission. So as many of you know, gay men are the most highly affected group by HIV in the US, and particularly young gay men have high rates of new infections, and we'll come back to that a little bit later in the presentation. About 70% of new infections in gay men are estimated to occur due to exposure of the rectal mucosa. And this is in part because of the high transmission probability of the receptive anal intercourse at 1.38% per exposure event. As we all know, the rectal mucosa is immunologically distinct from the female genital tract and the penis, and in large part likely because of the diversity of the microbiome, which is very relevant to this study as compared to other sites of mucosal HIV transmission, in addition to the single layer of columnar epithelium and the rich T cells that we find in the rectal mucosa. So our work in this area began a few years ago when we first compared the immunologic composition of the rectal mucosa among gay men compared to who were engaging in receptive anal intercourse compared to men who had never had receptive anal intercourse. And here you can see the global gene expression differences between MSM who are engaging in condomless receptive anal intercourse compared to controls in purple and green, and differences in the global gene expression based on timing of sexual intercourse. We found that pathways important in immune activation, cell proliferation, as well as mucosal injury and repair were enriched among MSM engaging in receptive anal intercourse. In addition, when we looked by flow cytometry, we found distinct differences in the CD4 T cell compartment as well as the CD8 T cell compartment among MSM engaging in receptive anal intercourse, and that they were enriched for Th17 cells, as well as proliferating CD8 positive T cells, as well as pro-inflammatory cytokine producing cytotoxic T cells, including both interferon gamma and TNF alpha. But we did not find any differences in CCR5, alpha 4, beta 7 expression in CD4 cells between MSM engaging or receptive anal intercourse versus controls. And when we looked at the microbiota by 16S rRNA, we, as well as other groups around the same time, found this very interesting enrichment for the Prevotella family over Bacteroides. And I think you can see here in these bar plots, this swath of yellow that represents the Prevotella family. Is enriched among MSM engaging and receptive anal intercourse compared to controls. We found other differences in the microbiome, in the composition of the microbiome, but these were to a lesser degree and of questionable significance, as they've not been replicated across other studies. Interestingly, we also found that relative abundance of Prevotella was associated with expression of the genes that were important in mucosal injury and repair. And so this got us to thinking about inflammation and the interaction of the microbiome as well in the rectal mucosal immune system and how those are related in inflammation. We've also looked at other biomarkers by immunohistochemistry, and here you can see we looked at some crypt epithelial markers. Here on this first panel is staining for E-cadherin, and then Ki67 in the next panel, looking at proliferation of the epithelium of the crypt. But we've also looked at lamina propria markers, including MPO, a marker for neutrophils, FOXP3, a marker for regulatory T cells that are often enriched in settings of inflammation in an attempt to suppress inflammation. And then we've also looked at IL-17 expression in the lamina propria of the rectal mucosa. This is actually not staining for CD4 IL-17 cells, but other IL-17 producing inflammatory cells in the lamina propria. And what's very interesting, if we measure these in a very systematic way and compare them between the two groups, as well as over time, is that we found no differences in FOXP3 expression or IL-17 expression between MSM and controls, although there may be a small difference in IL-17 as we go from the base of the crypt all the way to the lumen, where the site of exposure to HIV, other sexually transmitted infections and pathogens was thought to occur. We do see a suggestion of increased IL-17, although that did not reach statistical significance. We did find differences in MPO, or enrichment for neutrophil infiltration, among MSM engaging and receptive anal intercourse, as well as increased epithelial cell proliferation along the geography of the crypt, as well as towards the lumen of the lamina propria, being increased for MPO among MSM engaging and receptive anal intercourse. While there was some suggestion of E-cadherin being enriched among MSM, this did not reach statistical significance and was small in magnitude, but interesting nonetheless. Similar to our other studies where we did not find differences in or we did not find any associations between the microbiome and the cellular composition of the rectal mucosa, we also did not find any clear associations between expression of these biomarkers and the microbiome. The one exception to this is E-cadherin. We analyzed these data several ways, and while we did not find any associations in the global test of significance that I'm showing you here, there was some suggestion that E-cadherin was likely influenced by the relative abundance of Prevotella, although these findings are really nothing to take to the bank at this point in time because they were of marginal significance, but of interest nonetheless to follow up on. And so we've also tried to examine what is driving this enrichment for Prevotella among MSM engaging receptive anal intercourse. And we think that it may have to do with mucosal injury. And so this study we conducted was a clinical trial where we enrolled men who have sex with men into 3 different arms. In one arm, they took Truvada for 7 days. In one arm, they used hyperesmolar lubricant by the rectum for 7 days. And in one arm, they did both. And we measured the microbiome before and after after use of lubricant. In addition, we also measured PrEP drug levels to understand if PrEP drug levels could be influenced by the use of lubricant, but also whether the composition of the microbiome had any influence on PrEP drug levels in the rectum. And the hypothesis behind this study is the repeated use of hyperosmolar lubricant is thought to induce mucosal injury. And if that were true, we should see a shift in the microbiome away from Bacteroides and towards Prevotella. In addition, we know that the composition of the microbiota in the female genital tract is very important for PrEP drug efficacy, as has been seen in clinical trials, mostly in sub-Saharan Africa, where the composition of the female genital tract microbiome does influence those PrEP drug levels. And so we wondered whether this was also true in the rectum. And what we found was very interesting. In the two arms that used lubricant, which are the top panels and the bottom panels here, we did see a shift in the microbiome away from Bacteroides, with a significant decrease in the relative abundance of Bacteroides, as well as an increase in the relative abundance of the Prevotella genus. While this did not reach statistical significance, these two taxa tend to change in concert. One goes down while the other goes up, and so we do think that this finding is of significance. We also showed an increase in diversity of the microbiota associated with lube use, which is also of interest. Thankfully, we did not find any associations in the composition of the microbiota with PrEP drug levels, including tenofovir or emtricitabine. So there was no suggestion of that interaction that was seen in the female genital tract also being present in the rectum. And so this is very reassuring for the prevention efficacy of PrEP in interactions with the microbiome. So we think that condomless receptive anal intercourse exerts several mucosal immune effects through various kind of mechanisms. First, there's exposure to pro-inflammatory semen. There's direct microtrauma of the rectal mucosa to the rectum. And then we also get increased transient exposure of the lamina propria in the rectal mucosa to the gut microbiota. All of these exert several different immune effects that we've characterized, including an innate immune response that's likely mediated through neutrophils, potentially macrophages, and other inflammatory cytokines are likely involved as well. We've also shown evidence of an adaptive immune response, which includes enrichment for Th17 cells, CD8 cell proliferation, and cytokines, as well as that shift in the microbiota that I've been discussing that favors Prevotella over Bacteroides in men engaging in receptive anal intercourse. So the problem is this, with all of these data, while very interesting, is we don't really know whether this matters for HIV transmission or potentially response to prevention interventions. I've told you that there were no differences in PrEP drug levels in the mucosa, so that's good news. But what about response to other pre-exposure prophylaxis modalities, and even HIV vaccines, which likely would have differential response in inflammatory environments versus not? So how do we determine this? Well, that's all about study design in human studies. Ideally, we would have large longitudinal studies that have HIV incidence as their outcomes. These, as you know, are extremely expensive and difficult to do. I will tell you that recruitment to studies with mucosal sampling was difficult in 2019, and now with COVID, it's really just been turned on its head, and it's becoming increasingly challenging. The ground is shifting below us, and we're really having to relearn how we recruit for these studies. Mucosal sampling is just not acceptable to all people, and that's okay. So some proportion of people would not choose to participate in such studies. And there, of course, is some risk with mucosal sampling, mostly as we think of HIV transmission in the setting of damage to mucosal tissues after a biopsy procedure. So we've elected to try and determine whether we can use a proxy for HIV transmission in our laboratory. So our approach has been to define the human phenotype of interest, define that rectal mucosal immune milieu, and then use rectal explant challenges that were popularized by the Microbicide Trials Network to determine whether that can be a readout proxy of HIV transmission and replication in human tissues. And so we are currently conducting studies of adolescent MSM, and I'll show you some data about that, asymptomatic rectal STIs, and whether they have any influence on the mucosal immune environment, men who are having sex with and without condoms. So one question is if we would see the same results with and without condoms. And then finally, a study I'm incredibly excited about that we've just launched is to understand how feminizing hormone therapy in transgender women could potentially hinder or facilitate HIV transmission. And so that study is also ongoing. You'll see some pictures of how we conduct our explant challenges in ex vivo and tissue culture, and then the results as we challenge the tissue with HIV virus and then measure growth over an 18-day period. And what you'll see is that in between individuals, and these are 4 individuals that have very different trajectories of HIV replication in the explant challenge model. And can we use this trajectory or this area under the curve as a marker for HIV susceptibility or potentially HIV replication, which are actually 2 different questions, in our studies to help understand human differences in HIV transmissions. And so what this has allowed us to do, just as an example, we've characterized extensively the innate immune cell subsets in the rectal mucosa. And here I'm showing you the abundances of these innate immune cell subsets, including B cells, natural killer cells, gamma delta T cells, mate cells, dendritic cells, macrophages, and neutrophils, and shown here in decreasing abundance within rectal mucosal tissues. We're able to characterize the abundance of these cells by individual, and then determine whether there is any association with HIV replication in the rectal explant challenge model. And interestingly, we found that marginal zone B cells were positively associated with HIV replication in the explant model, challenge model, while natural killer cells that exhibit a lot of antiviral properties, the relative abundance of those was negatively associated with HIV replication in the explant model. None of the other cellular subsets were associated, or innate cellular subsets were associated with HIV replication in the explant model. But this has given us a lot of preliminary, very interesting data to then follow up on to understand how neutrophils and potentially marginal zone B cells are operating within the rectal mucosa at the time of HIV exposure and transmission to either facilitate HIV transmission or potentially exert antiviral effects in the case of natural killer cells. In addition, we've also been able to look at adolescent gay men to understand if the rectal mucosal immune milieu differs between adolescent and older MSM. And these differences, we hypothesize, are not only due to chronological age, but also likely less lifetime exposure to receptive anal intercourse. And you can see here, we've enrolled 2 groups, 32 adolescent MSM versus 34 older MSM. The adolescent MSM were aged 18 to 21. The older MSM were aged greater than 35, and we were successful in recruiting adolescent MSM that had an overall lower number of receptive anal intercourse partners in their lifetime compared to older MSM. In addition, a lower number of receptive anal intercourse encounters in the last 12 months among the adolescent MSM versus the older MSM. And so these populations were skewed not only by their chronological age, but also their lifetime behavioral exposure to receptive anal intercourse. And so when we compare these 2 groups with flow cytometry and look at the CD4 T cell subset composition, the CD8 T cell composition, as well as the innate immune cell subsets, we do find some interesting differences. So just to remind you, the adolescent median age group was 20 years of age, while the older MSM was quite a bit older at 46 years of age. And what you see is that among the adolescent MSM in the CD4 T cell compartment, that it was enriched for naive CD4 T cells compared to the older MSM, as one might expect. No differences in the resident memory CD4 T cell population. Interesting, there was a very significant reduction in CCR5 expression among the adolescent MSM compared to the older MSM. However, an increase in Ki67 or proliferating T cells. So naive proliferating T cells are more abundant among adolescent MSM in the rectal mucosa compared to older MSM, while CCR5 expressing CD4 cells are more prevalent in the older MSM. In addition, cytokine positive, both CD4 and CD8 T cells were increased among the older MSM, decreased among the adolescent MSM. In addition, compared to older MSM, adolescent MSM had lower levels of CD8 tissue resident cells. We did not find any clear differences in the innate immune cell subsets when we compared adolescent versus adult MSM. And this is something that will require further analyses, but there's no clear differences in the 2 groups here. And then when we look at HIV replication in our explant model, we do see a significant difference among the adolescent or young MSM compared to the older MSM, with younger MSM exhibiting higher levels of HIV replication in our explant model, significantly higher than adult MSM. And when we look to see whether any of the cell subsets, some of which I've already told you about it, were positively or negatively associated with the area under the curve or HIV replication in the explant model, we did find that Ki67 CD4 positive T cells or proliferating CD4 positive T cells that are enriched among the young MSM are positively associated with HIV replication in the explant model. Marginal zone B cells also positively associated. I've already shown you that data. In addition, CD4 cells that produce antiviral cytokines such as TNF and interferon gamma, also pro-inflammatory cytokines, were negatively associated with HIV replication in the explant model. We're currently investigating whether these CD4 positive T cells also produce other antiviral cytokines that could be a potential explanation or mechanism for that reduction in HIV replication in the explant model. And I've also shown you the difference or the reduction associated with NK cell prevalence in the rectal mucosa with HIV replication. So very interestingly absent from these associations, again, is CCR5. And CCR5 is very abundant. Expression on CD4 T cells is very abundant in the rectal mucosa and obviously necessary, absolutely necessary for HIV infection and transmission. However, in our findings, it may not be that it is the most important factor, but the most important factor really proliferating CD4 T cells that support early replication in the rectal mucosa. And so we are still kind of working on teasing out to all of these differences. Since this is a kind of microbiome conference, I had hoped that we would be able to show you some additional data on differences between adolescent and adult MSM. And unfortunately, COVID-19 has resulted in significant delays in our data kind of acquisition and analyses, and we are just now getting those kind of back online. But we do see some global differences between the microbiota comparing adolescent versus adult MSM in preliminary analysis. And in addition, we do show that adolescent MSM appear to have the highest Bacteroides versus Prevotella ratio when comparing to older MSM and men who don't engage in anal intercourse. We're continuing to work on these analyses and fine-tuning them such that hopefully we will have more data to share with you in the near future. But I do think that these are going to be very interesting findings to follow up on. So I will go ahead and wrap up there. I'd like to extend a very heartfelt thank you to our participants. We ask a lot of our participants to come in for mucosal sampling into our clinic and undergo these procedures. So we are always so thankful when they agree to participate. In addition, our laboratory, our collaborating labs, of course, and our funders. I want to say thank you very much.
3, 2.
Thank you so much, Dr. Kelley. That was a fascinating talk. I really enjoyed that immensely. So our next speaker is Jason Brenchley. Jason Brenchley is a senior investigator and chief of the Barrier Immunity Section in the Laboratory of Viral Diseases within the intramural program of the NIH. His work is focused on understanding the causes and consequences of unique pathologies that occur within the GI tract that occur with HIV-infected individuals. Dr. Brenchley has published over 150 peer-reviewed manuscripts focused on inflammation, mucosal immunity, and the microbiome and HIV pathogenesis. And today, he's going to talk to us about his work on the microbiome and SIV acquisition.
All right, well, I'd first like to thank the organizers for the opportunity to present some of our recent work related to how the composition of the gastrointestinal microbiome can contribute to progressive SIV infection of rhesus macaques. I'm a federal employee. I think everyone knows that. As such, I never have potential conflicts of interest to discuss. Just as a little bit of background, I think everyone in this room is very well aware of the data that have shown that cross-sectional analysis of the composition of the gastrointestinal microbiome between HIV-infected and HIV-uninfected individuals routinely results in the demonstration that the HIV-positive microbiome is altered compared to population controls. But when we've looked longitudinally in SIV-infected Asian macaques, we really don't see evidence that the composition of the microbiome is becoming dysbiotic as a result of SIV infection. And so kind of compared to what people have done in cross-sectional studies of HIV-infected patients compared to HIV-uninfected controls, these are the same animals in the same cage, getting the same base diet. And what we're looking at here is the composition of the microbiome in 11 animals pre-SIV infection, and then during the acute phase of SIV infection when inflammation is really going rampant. In the green, we have the Bacteroidetes. In the pink color, we have the firmicutes. And in the blue are the proteobacteria, which include a lot of the pathobiotic bacteria that we think of as being contributing negatively to the composition of the gastrointestinal microbiome. There's a large portion of the Bacteroidetes that are Prevotella, which is seen kind of to be expanded in HIV-infected individuals. But overall, if you average the microbiomes of the animals pre-SIV infection and during day 21, you really see no evidence that there is any alteration to the composition of the gastrointestinal microbiome as a result of SIV infection. And this is based upon analysis of 16S data. You start to see subtle differences if you do shotgun sequencing, and maybe Roger Paredes is going to talk about that. And in fact, Roger has shown that if you kind of control for risk factors that led to people becoming HIV infected, it's still doing cross-sectional analysis, but it seems like risk factors that lead to people becoming HIV infected themselves are associated with alterations to the composition of the gastrointestinal microbiome. So it really becomes a question of whether or not HIV itself is changing the composition of the microbiome. But analysis of the microbiome by sequencing is really just telling us who is present and who is not present. The 16S analysis doesn't really tell us anything about the functionality of the gastrointestinal microbiome. And so one of the things that we've been doing is trying to isolate and identify bacteria that are actually translocating from the lumen of the gastrointestinal tract into peripheral tissues. And what we're looking at here is the identity of microbes that we're able to actually viably isolate from liver, from mesenteric lymph node, and from spleen of chronically SIV infected rhesus macaques. If we do the same type of analysis in SIV uninfected macaques, we rarely find any bacteria that are viable. And the red bacteria are predominantly firmicutes. We've actually tried to grow bacteria anaerobically, and in a couple of cases have been able to find a few Bacteroidetes. And the blue bacteria belong to the proteobacteria phylum. And so what we're seeing is that there's a lot of firmicutes and a lot more proteobacteria than are present in the gastrointestinal microbiome. And so there does seem to be kind of a preference for proteobacteria translocating. In collaboration with Adam Burgener, what we've been able to do is to look at the proteomics of the individual bacteria that have translocated and compared the proteome to bacteria that are still from the lumen of the gastrointestinal tract of rhesus macaques, but are not translocating. And you can see here that there's a lot of proteins that are being differentially expressed in the bugs that translocate versus the bugs that don't translocate. And if we do some sort of pathway analysis, what we find is that there's really one specific protein that is expressed by 5 of the 8 translocating bacteria that we studied, and 2 of them being proteobacteria, and 3 of them, Lactobacillus, Weissella, and Kurthia, being firmicutes. So even though these are very diverse bacteria belonging to different phyla, 5 of 8 of them are expressing the cytosine-specific methyltransferase. And so what we're trying to understand now is whether or not expression of this methyltransferase is actually influencing the ability of bacteria to translocate to different anatomical sites in chronically SIV-infected macaques. But one of the questions that we really wanted to try and address is now we don't think that significant dysbiosis is happening as a result of SIV infection of Asian macaques. But the question is, well, what if we induce dysbiosis? Does that influence any aspect of progressive infection, or does it change the susceptibility of an Asian macaque to acquire SIV? Our hypothesis is really that in the context of increased microbial translocation, which we know is already caused by progressive HIV and SIV infection, if we induce on purpose and experimentally dysbiosis, we could actually further increase microbial translocation, which we think would then lead to enhanced systemic inflammation, which would enhance loss of GI tract resident and peripheral CD4 positive T cells, and of course, accelerate SIV disease. So these data were published a couple of years ago, so I'm just going to show a couple of slides to highlight what our findings were. We used vancomycin to induce a significant dysbiosis in SIV uninfected Asian macaques. And what we're looking at here are several different animals that are color-coded, and the circles are the squares are pre-vancomycin and the circles are post-vancomycin, and you can see that the first principal component here is really separating the animals based upon vancomycin treatment. The second principal component is separating the animals based upon identity. So the purple animal is up here, the green animal is here, the orange animal is here, and so on. So vanc is really inducing a significant amount of dysbiosis. And that dysbiosis looks kind of similar to what people have observed in HIV-infected individuals, with kind of a collapse of the firmicutes. And this chronoplot, we're looking at the residual firmicutes that exist. Here are the control animals, here are the vanc animals. You see a significant reduction in the amount of clostridiales in the vanc-treated animals, and you see a significant expansion of proteobacteria. And of the proteobacteria in controls, you see a lot of proteobacteria that are kind of diverse. You see alpha, beta, epsilon, gamma. But in the vanc-treated animals, you see predominantly a huge expansion of the gamma proteobacteria, which include a lot of the things that we talked about earlier that we think of as being pathobiotic. So the question was, if we induce this significant dysbiosis in SIV uninfected macaques and then infect them, do we accelerate SIV disease? And one of the things that we first looked at was kind of the functionality of the gastrointestinal microbiome. We looked at short-chain fatty acids. We looked at valeric acid, which is a 5-carbon short-chain fatty acid, and hexanonic, which is a 6. Both of them were significantly reduced because you get rid of a lot of the firmicutes, and in particular the clostridiales. The loss of the short-chain fatty acids resulted in damage to the structural barrier of the gastrointestinal tract. So this is a chronically SIV-infected macaque, a colon section, an immunohistochemical analysis of tight junction protein claudin is in brown. This may be a focal area of damage to the structural barrier of the gastrointestinal tract, might be an artifact, but overall the epithelial barrier is pretty intact. And you contrast that to what happened in the animals who got treated with vanc, you see significant destruction to the structural barrier of the GI tract, a lot of infiltration of leukocytes, which would be consistent with inflammation. And when we do quantitative image analysis, you see that there's significantly more destruction to the gastrointestinal tract in the vanc-treated animals compared to the controls. But surprising to us, the animals who were treated with vanc progressed to AIDS at the same rate as the animals who were SIV infected but not treated with vanc. So even though there was a significant destruction to the gastrointestinal tract epithelium, this did not accelerate SIV disease. And so one of the things that we were then wondering is, well, how much inflammation does dysbiosis by itself cause? So again, this is looking at the composition of the gastrointestinal microbiome before vanc and after vanc, and you can see significant induction of dysbiosis in the vanc-treated animals. And now what we're looking at is how much inflammation does that vanc by itself induce? If we're talking SIV uninfected animals, pre-treatment with vanc, and then post-treatment with vanc, and we're looking at the frequency of activated CD4 positive T cells that express Ki67. And you can see in all 4 animals, you see a slight but statistically significant increase in the frequency of activated CD4 positive T cells that are proliferating. So knowing that there is significant destruction to the epithelial barrier and activation of CD4 positive T cells, we thought maybe easier for the animals to acquire SIV. We also went on to kind of look more globally at how much inflammation was induced by vancomycin treatment in non-human primates that were SIV uninfected. And what we did is we did gastrointestinal biopsies, colon biopsies, and then we did transcriptional profiling by nanostring, which is a targeted reverse transcriptase PCR of about 800 genes of immunological interest. So it's kind of a cheap way of doing a transcriptional analysis. So what we're looking at here is a PCA plot, dimension 1 versus dimension 2. The purple circles are pre-vanc, the blue circles are post-vanc, and you can see that globally there wasn't really a significant alteration to the immunological transcription profile of the colonic cells before or after vanc. But we did kind of look at genes that were differentially regulated. There were about 29. And if you do pathway analysis on that 29 genes, the first pathway that lights up are toll-like receptor signaling. So we do think that just by itself, vancomycin is inducing low level of inflammation. The next question we wanted to ask is whether or not the dysbiosis that we induced, does that make it easier to acquire SIV? And so we started doing low-dose interrectal challenges in animals that were treated with vanc versus controls. And we used Brandon Keel's barcoded SIV MAC239 so that we could look at the number of transmitter founder viruses. So this is a virus called SIV MAC239X. What Brandon does is introduce barcodes into the polymerase gene, and they're synonymous mutations. So it doesn't change the functionality of the virus. It just allows us to be able to identify the number of transmitter founder viruses. And Brandon has shown that each one of these barcoded viruses replicates efficiently and kind of equivalently both in vitro and in vivo. And so what we started doing was low-dose interrectal challenges in animals that got vancomycin. We titered the virus such that about every time 20% of the animals would get infected. And you can see after 10, it's actually after 11 challenges, all of the control animals got infected. We then treated animals with vancomycin for 1 week, then challenged them with SIV MAC239X and looked at the number of challenges that it would take to infect all of them. And we went down to about 6. So after 6 challenges relative to 11, we infected all of the animals with SIV MAC239X after being treated with vanc. So this didn't really achieve statistical significance. It was close. But what we were able to then look at is how many transmitter founder viruses there were after the animals actually got infected. So you can see here are the controls, the number of transmitter founder viruses on the y-axis, and all but one animal that was a control got infected with 1 and only 1 transmitter founder virus. 1 animal got infected with 2. Now, if we contrast that to the animals who got vancomycin, you can see that 4 of them got infected with 1 and only 1 virus, but 1 of them got infected with all 10, another got infected with 6, 5, and 2. And so that ended up being significant, statistically significant, that there were more transmitter founder viruses in the vancomycin-treated animals compared to the controls. So just to kind of wrap up, vanc-induced dysbiosis is insufficient to accelerate SIV disease progression. Vanc-induced dysbiosis is sufficient to induce low level of GI tract inflammation. This barcoded SIV MAC239 can be used to assess the number of transmitter founder viruses and the proclivity to become SIV infected intrarectally. And dysbiosis that we induced by vancomycin results in increased susceptibility to acquisition of SIV with higher numbers of transmitter founder viruses. So we know that risk factors for HIV acquisition are associated with a dysbiotic microbiome. And so we kind of think that maybe therapeutic interventions to reduce dysbiosis may decrease susceptibility to HIV acquisition via receptive anal intercourse. So I'd just like to close there and thank all of the investigators who were important for this work to be done. First and foremost, Alex Ortiz, who is a phenomenal staff scientist in my lab that really led the projects on how dysbiosis influences disease progression and influences susceptibility to SIV acquisition. Jacob Flynn is a graduate student in the lab who has been growing bacteria from different anatomical sites and categorizing and characterizing them by proteomics with the help of Adam Burgener that I mentioned earlier. All of the microbiome work that we've done is in collaboration with Yasmin Belkade with Health Bioinformatically from Marion Quinlan's. I mentioned that I had the SIV MAC239X from Brandon Keel at NCI Frederick. I think everyone knows that the immunohistochemical analysis that I present is always done in collaboration with Jake Estes, and none of these studies would be possible without a very dedicated animal care staff at the NIH Animal Center. And I'd finally like to thank you for your time and attention and be happy to take any questions.
Thank you so much to all three of our speakers for really excellent and thought-provoking talks. So at this point, we have 15 minutes for a discussion of all three talks. And we already have a few questions that have come in the live chat, but all attendees, please feel free to keep typing in your questions over the next 15 minutes while we have this discussion. So I'm going to kick off the discussion with a question from Kara Wilson. And this question is for Dr. Kelley. And Kara's asking, an altered Prevotella Bacteroides ratio has been observed in the sigmoid colon as well as in the rectal mucosa in MSM. Since direct mucosal injury is less likely to explain these colonic changes, what mechanisms might promote the rectal dysbiotic phenotype throughout the colon?
Well, I mean, first I would point out that it's not a dysbiotic phenotype to have enrichment for Prevotella. I think that's something we need to carefully delineate between what actually is dysbiotic and isn't. It can be representative of a very healthy microbiome. So that's one small point though, but it's a little bit of a sticking one for me. But anyways, I think it's a great question. I think we don't know. I think that when people have studied receptive anal intercourse with sham sex and things like that, they do find that kind of forward, what's the word? I guess more proximal effects can be seen with semen being transferred further up. I think you're right in that one would hypothesize that direct mucosal injury doesn't occur at those more proximal parts of the colon, but I would imagine that you might see fluctuations in the microbiome further along than where the direct injury occurs. I will say that others, I think our host group or our moderators group has seen the same thing in men MSM who do not report receptive anal intercourse, but only report being the insertive partner. We anecdotally have seen the same thing, although I think that's a little bit complicated by desirability bias when you're reporting your behaviors. So I think there remains more to be explored. I think that our lubricant use definitely shows that can be one study, can be one contributing factor, but there's likely many others that should be teased out. So hopefully that answers the question.
Well, thank you to our speakers for the beautiful presentations. I think some people in the audience want to replicate Dr. Kelley's experiment. There are two very specific methodological questions. One is from Elizabeth Menu: which HIV strand did you use in your rectal mucosal explant model? And there's also Yukin Choi asking about if you use fresh biopsies or frozen biopsies.
Fresh, I don't think it would be possible with frozen, although we've never tried. We've used BAL just because it's easy and it's pretty standardized, but are hoping to add in transmitted founder variants as well into our experiments. They're not as reliable when you're looking at comparing, I think, between groups of people, but we're hoping to make that transition for future studies.
Great. So we have a question for Dr. Burgener from Lindy Mason. And Lindy is asking, were you able to stratify into L-inners and non-inners lactobacillus groups? And did you see different effects of DMPA on HIV in these groups?
Thanks for the question, Lindy. It's good to hear from you. That's a great question. So we did a sub-analysis stratifying women based on L. crispatus or lactobacillus inners, dominant microbiomes. With Lactobacillus inners, they made up the majority of women in CAPRISA 004, and the Lactobacillus dominant type. And we saw the same observation. Within the small group of L. crispatus women, we couldn't do a case control analysis because all of the cases, there were 3 or 4 of them, all of them were in the DMPA group. So there was no way to look at relative risk. It was just too small of a group to do a sub-analysis. But within the Lactobacillus inners group, we saw the same observations as we did for the entire all Lactobacillus dominant women.
Okay, we have many interesting questions. Another one from Lindy Mason to Jason Brenchley. Jason, do you have a hypothesis for the mechanism by which the material transferase might influence translocation?
Yeah, we were trying to understand whether or not the methyltransferases being differentially expressed have an influence on the functionality of the microbes that we isolate. So methyltransferases are kind of known to influence the epigenetics of the host. And so the way that we're trying to address this is twofold. One is to try and sequence the epigenome by sulfite sequencing. And so we're doing that in the bugs that translocate versus the bugs that don't translocate. And then there is a methyltransferase inhibitor that can be used in vitro, and we're trying to use that to understand whether or not interfering with the methyltransferase influences at least the growth kinetics of the bugs that we've identified that have translocated. But that's kind of our working hypothesis, so stay tuned.
What do you think accounts for the difference between humans and macaques in terms of HIV/SIV impact on the microbiome?
Yeah, I was kind of hoping that that question wasn't going to get asked. This is definitely controversial, but very reproducible. There have been several groups that have used 16S sequencing to look at the composition longitudinally in non-human primates before and after they acquire SIV. And really, unless you look in fulminant AIDS, do you see any sign of alteration of the composition of the microbiome in non-human primates. If you look acutely, like I showed, there's really no difference to the composition of the microbiome. Into the chronic phase, the microbiome stays pretty constant. But when animals progress to fulminant AIDS, you absolutely do see some differences in the composition of the microbiome. So obviously the main advantage that we have in the non-human primates is it's the same animal in the same cage getting the same base diet. So we're trying to minimize the number of confounding variables that could be contributing to the composition of the microbiome. The downside is that we don't have nearly the numbers that we can do with humans. We can only study a limited number of non-human primates longitudinally. But I think it's probably mostly confounding variables that are influencing the composition of the microbiome in HIV-positive people, at least while they're asymptomatic. And there have been several groups that have shown that, including Roger's group. We heard today from Kelley or from Colleen that was happening. So I think it's mostly confounding variables. And I'm curious to hear what other people have to say on the topic, but that's my thought.
Another mechanistic question for Dr. Kelley from Cara Wilson in Colorado. An altered Prevotella bacteroides ratio has been observed in sigmoid colon as well as in rectal mucosa and MSM. So since direct mucosal injury is less likely to explain these colonic changes, what mechanisms might promote the rectal dysbiotic phenotype throughout the colon?
I think we already did that one. Although it is my favorite topic to talk about, we'll maybe go to another one.
Well, I'll ask you a different one, Dr. Kelley. So I had a question. When you were doing your assays, the ex vivo, and finding immune populations, and found some things like proliferation markers were influenced, but you brought up how CCR5, you were surprised to not see that one come up. And I was just curious, we thought about this in our similar work sometimes, about a bit of two things going on where cells, they're more likely to be infected, but then they're killed by HIV. And do you think that, that could, like for instance, correlations in biopsy, sometimes we'll see stronger correlations of microbes with a CCR5 expressing T cell in a non-HIV infected person, and then that kind of goes away in HIV. And we just wonder if it's because it gets, those cells get killed once they're infected. So I was just wondering if you thought that could play into that kind of negative result at all.
Yeah, I mean, but what we're looking at is at the HIV negative time point, the pre-challenge time point, and then associating that. I mean, honestly, I think probably what it is all the T cells in the rectum are CCR5 positive, and so you're not going to necessarily get a readout when you have such a high abundance or a high prevalence of CCR5 that it's actually those all other characteristics of the cells. Are they activated? Are they proliferating? That make them most susceptible. That's what I think. But maybe others that have more expertise in that might have other ideas.
Adam, a question for you. If you had to forecast the future of the vaginal microbiome and the risk of HIV acquisition, which will be your bet in terms of defining a therapeutic target to decrease the risk of HIV acquisition? Will that be dysbiosis, a bacterial species, a metabolite?
I'm sorry, what specifically was the question? What do I think the future of therapies for resolving bacterial vaginosis, or?
Yeah, to decrease the risk of HIV acquisition in women, if you had to define a target to design a clinical trial, what do you think?
That's a fantastic question. There have been many trials looking at probiotics to reduce the incidence of bacterial vaginosis, the recurrence of bacterial vaginosis in the context of metronidazole therapy, and some of them had some success, but it's never 100% success. So it's definitely clear that we're missing some key features of reconstitution of, if we're trying to promote vaginal lactobacillus, we're missing some key components that might be important. And trials from vaginal microbiome transplantation have shown some really nice early results of these really small trials. And they contain all components of the mucosa, since these are sponges transplanted from one woman with lactobacillus to another woman that was being treated for a BV. And it perhaps that mix of metabolites and bacterial species may be more successful in resolving. But I don't have the answer on which specific component is important for resolution of bacterial vaginosis. That is the very important question.
I have another question actually related to translation of your research, Dr. Burgener, and that was sort of that, I thought it was an interesting finding that contraceptive, DMPA, was it was only detrimental in one microbiome type, the Lactobacillus microbiome type. So is there sort of a future there for personalized medicine that, you would only give that contraceptive to someone who didn't have that microbiome type? But I just think you hit challenges with, in practice, actually characterizing the microbiome in low resource settings and things like that.
These are really great questions. I don't think that this one research study has any implications on policy and recommendations for which type of contraceptive is. I think that's up to the individual health provider in discussion with their patients. But so whether or not I think that this could inform contraceptive choices, I don't know. I think that this study is hypothesis generating, since it's a post hoc analysis, and I don't think we should be careful not to overinterpret the results. But I think that is a question that needs further study to see if it's validated in other cohorts or in other settings or other trials. But I would not I do not think that it's too early to say that this would be important for informing women who are at risk for HIV.
I think we have time for one last question from Mariangela Cavarelli for Jason Brenchley. Does the microbiota return to baseline levels after vancomycin treatment interruption?
Yeah, so the Animal Care and Use Committee would only allow us to give vanc for a week. So we gave vanc for a week, then we do a washout, and then the next month we give vanc again. And the microbiome does not maintain the same degree of dysbiosis that you see acutely after vancomycin treatment. Does it return to what it was before? More or less, yeah, within the month that we start initiating the next round of vanc, it's back to a pretty good level of what normalcy looks like. But if you follow an animal's microbiome, even though it's the same animal in the same cage in the same diet, month 1 versus month 2 with no intervention, the microbiome is not completely static. So I think it's kind of impossible to know if it came back to exactly what it was supposed to be, but yeah, it does heal itself, which is why we kind of continued to give vanc. And with the low dose and rectal challenge study, we would give vanc for a week and then the next Monday do the challenge. And then if they got infected, they were done. If they weren't infected, then we would wait 3 weeks, give vanc again, and then challenge on the next Monday. So we would try to make sure that we were inducing dysbiosis to see if it would influence acquisition.
Okay, so we had more questions, really interesting from the audience. Unfortunately, we have to close the session. Thank you very much to our speakers for the wonderful talks and to the assistants for their engagement. Just to wrap up the session, Dr. Burgener has shown the relationships between oral contraceptive, vaginal microbiota profiles associated with increased genital inflammation, the risk of HIV acquisition. Dr. Kelley has defined the rectal immune profiles associated with higher HIV infectivity using explant experiments with rectal mucosa from adolescent and older MSM, and showed the differences in the mucosal immune subsets between these two groups. And Dr. Brenchley has shown us different mechanisms by which dysbiosis can promote mucosal inflammation and SIV acquisition, the potential therapeutic interest of the microbiome. So I think we can close the session. Please don't forget to complete the evaluation form. This is really important for allowing us to improve the next HIV and microbiome workshops. And we really hope to see you in the next session. Thank you very much.